HubLE community. My name is Dr. Ramchand. I am an endocrinologist and clinician researcher at the University of Melbourne. I am currently undertaking a research fellowship at the Mass General Hospital with Dr. Lena, looking at combination therapy for osteoporosis. This year's ASBMR meeting, as many of you know, was a virtual meeting, and while we could not meet each other in person, it did not disappoint on the science. We had an amazing meeting with fantastic data presented. One of our young investigators, Dr. Lena Batoon, that is with me today, gave an oral presentation at this meeting. Lena, would you like to introduce yourself further, please?
Hi, I'm Lena Batoon, and I am a final year PhD student at Mater Research Institute and the University of Queensland under the supervision of Professor Alison Pettit.
Perfect. Would you like to tell us what you presented today, presented at the meeting?
Sure. So I presented on the bone phenotype of the CSF1R knockout rat model on a defined genetic background. So CSF1R stands for the colony stimulating factor 1 receptor, and CSF1R signaling controls the proliferation, differentiation, and survival of macrophages and osteoclasts, which are two cells that have very important roles on bone.
So Lena, that was a fantastic presentation. Would you like to tell us a little bit more about what the key findings were and the bigger picture of your research?
Yeah. So using the CSF1R knockout rat model, our lab was able to demonstrate biologically distinct roles for osteoclasts and macrophages during skeletal development and somatic growth. In particular, we showed that macrophages have more important roles during bone development than originally thought. We were also able to rescue the skeletal phenotype in our CSF1R knockout rat model using wild-type bone marrow transplantation, which exposes the therapeutic potential of targeting the CSF1R axis in related bone diseases. So CSF1R mutation in humans is actually associated with neurodegenerative disorder, mild to severe osteopetrosis, and even early infant lethality. So by being able to rescue the phenotype in a rat model by just wild-type bone marrow transplant, our findings demonstrate that the severe phenotype in patients with CSF1R mutation can almost be entirely reversed, which is very promising.
Yeah, that would be fantastic. And so what's next for you? What's next for the study?
Well, I'm hoping to submit my thesis in 3 months in December, and I'm currently scoping for postdoctoral positions. I actually had plans to go to the US to visit 5 labs there, but unfortunately because of the current pandemic, I had to cancel those plans. So I'm currently scoping postdoctoral positions here in Australia, and I still hope when work on bone research.
I'd like to thank you so much for your time today. Congratulations again on your talk. It was fascinating, really, really good work. And we look forward to meeting you again in person at next year's ASBMR meeting. So thank you very much.
Thanks so much for having me.